The next speaker will be Sonja Boland, also on the respiratory tract, but with different particles and different questions, and I hope answers.
Yeah. Thank you. Good afternoon. I will talk you about our results on the effects of carbon black and titanium nanoparticles on respiratory epithelial cells. And I will show you that these particles induce the same effects, but through completely different mechanisms. So our objective was to study the effects of nanoparticle on respiratory epithelial cells, as these are the first targets after inhalation of nanoparticles. So firstly, we studied whether these particles are taken up by the epithelial cells and if they could induce the production of reactive oxygen species. As has been shown that the ROS inside the cell could lead to antioxidant defenses or the induction of a pro-inflammatory response. And at very high concentrations of oxidative stress, this could even lead to cell death. And I will show you here, we have studied all these three endpoints and the mechanisms involved, but here I will only show you the mechanisms involved in the induction of cell death. We used the bronchial epithelial cell line 60HBE and we confirmed the results with primary cultures. And we used these two types of nanoparticles, carbon black and titanium dioxide. And we used the different sizes of nanoparticles within the nanometer range. So transmission electron microscopy is showing you here that these particles are taken up by epithelial cells and they are present in vesicles, but sometimes they could also be present free in the cytoplasm without a membrane around. And this is mostly seen with the titanium nanoparticles. And both types of nanoparticles are mostly forming aggregates, which are quite loose, but could be very important agglomerations. And sometimes you can also see individual or very small aggregates, which are taken up by the cells. And it's mostly these small aggregates, which could even enter the nucleus, especially for the carbon black nanoparticles. These two types of nanoparticles induce the production of reactive oxygen species, as we have seen here by the oxidation of the dihydroetidium dye by flow cytometry. And you can see that this effect is size dependent, as the smallest nanoparticles have the highest potential to induce oxidative stress. So you can see that even though these particles are present in aggregates, the final biological effect is still dependent on the primary particle size. And there is a very good correlation between the oxidative stress induced and the specific surface area of the nanoparticles. And this correlation is even is better for the surface area than the primary particle size, as this takes into account also the porosity of the particles. So this is really the surface which is present, which can induce the responses. So then we studied the induction of cell death by FDA ethidium bromide staining, and you can see here that there is dose-dependent induction of cell death for both types of nanoparticles. And the quantification of apoptotic cells is showing that this cell death is due to an increase of the apoptotic cells, but not the necrotic cells, whereas the positive control H2O2 is inducing both types of cell death. So to further analyze the mechanisms involved in this cell death, we studied the effect at 20 microgram per square centimeter. And we confirmed by transmission electron microscopy that there is an induction of apoptosis. As you can see, characteristic morphological changes, such as chromatin condensation here, as you see with both types of nanoparticles, and membrane bleving. This induction of apoptosis was further confirmed by the measuring of the DNA fragmentation by quantifying the sub-G1 peak in flow cytometric analysis. And both types of nanoparticles induce the activation of the caspase 8, which is the initiator caspase, and the effector caspases 3 and 7. However, there's only the carbon black nanoparticles induce a depolarization of the mitochondrial membrane potential, as revealed by an increase of the staining of, with the CMX-ROS dye. And this induction, a mitochondrial pathway is confirmed by immunohistochemical analysis, showing that there is a release of cytochrome C from the mitochondria, but only after carbon black treatment and not with titanium. And there is an activation of the pro-apoptotic protein bags. So carbon black particles induce the apoptosis via the mitochondrial pathway, and this is ROS dependent, that all these events could be inhibited if you use a catalase. And this, the catalase has no effect on the titanium induced cell death. On the other hand, titanium is inducing lysosomal destabilization, as you can see here that there is an increase of the acridine orange staining of the cells, and this is also confirmed by release of the cathepsin B from the lysosomes, and cathepsin Bs could activate caspases, inducing cell death. So this destabilization of the lysosomes could be due to membrane damages, which could be induced by lipid peroxidation, for instance, and indeed we can see an increase of the lipid peroxidation marker body P by the titanium nanoparticles. So titanium is inducing lipid peroxidation, lysosomal membrane destabilization, and activation of caspases leading to apoptosis. So in final conclusions, I've shown you that there is a contribution of the primary particle surface area of nanoparticles in the biological responses, even though these particles could be aggregated in the cell culture. So there is a very good correlation of oxidative stress with the BET surface, and we have also seen this for the pro-inflammatory response, for instance. On the other hand, we have shown that there's a comparable effect of mineral and combustion-derived nanoparticles, which induce the same cellular responses, but through different pathways, as I've shown you that the apoptosis is induced by the mitochondrial pathway for carbon black, and the lysosomal pathway for titanium. So it's very important also to know which mechanism the nanoparticles induce this effect, not only the final endpoint, because if you want to produce nanoparticles safe by design, it's easier to know really why and how these particles react. So finally, I want to thank all the people who worked on this subject, and the platforms we have used, and the finances. And I would especially acknowledge Sadiq Hussain, who has made this during his thesis. So thank you for your attention.
Thank you, Sonja. We have time for one or two questions. So who wants to ask a question to? So I have a question for you. For the lysosomal pathway, did you see any indication that you may have autophagy of the system, because it's a
no, we just for the electron microscopy we didn't see it, but we didn't test any. We didn't go further and analyze it if there could be some autophagy.
But you have seen the release of cathepsin B in the cytoplasm, not outside of the cells?
Yeah.
They didn't secrete cathepsin B?
We didn't look on the secretion. We just, this was just immunohistochemical detection of release of the cathepsin B from the lysosomes, but it was detected inside the cell.
Okay. Other question?
There's apoptosis that turns on the immune system, and then there's apoptosis that doesn't turn on the immune system. Do you know whether the two forms of apoptosis that you're getting would, for example, activate macrophages or dendritic cells that were mixed in with your epithelial cells?
If these cells could, I'd most say that there could be link because to inflammation because of the inflammasome we have seen this morning, as cathepsin could also. I've shown that there is an activation of caspases, and caspases are involved in the inflammasome complex. So and we have seen that there's some immunoresponses to these particles. So this
To both of them.
could be.
To both of them.
Yeah, we have seen a cytokine induction, but the problem is with the nanoparticles that the cytokines get absorbed onto the surface of the particles and you can't detect them anymore. So for IL-1 beta, for the inflammasome, we were more looking on the mRNA expression. But the inflammasome, you have to look on the secretion of the major protein. So this we couldn't do because of this interference with the test system. So we have maybe to look on other techniques to see if there is inflammasome induction by these particles. It's more a guess because if it's inducing caspases, then maybe they induce also inflammasome.
Other question?